Touch a hot surface and you withdraw your hand instantly, before you consciously think about it. And thanks to the lightning speed of your nervous system, you may escape with mild discomfort rather than a third-degree burn. In this lecture, we'll explore how nerve cells, also known as neurons, handle information so rapidly. We'll start with a look at the distribution of nerve cells in our bodies and how they're organized. Then we'll examine how signals pass from neuron to neuron, and from neuron to muscle cells using electrical signals and biochemical molecules called neurotransmitters. Some molecules, such as dopamine, that play roles in passing on neural information, also function as hormones in the bloodstream. But neurotransmitter signaling is completely different. Fast-acting hormones like adrenaline take minutes to act, and slower-acting steroid hormones like cortisol may require hours. By contrast, neurotransmitters send signals that work in fractions of a second. The difference is akin to sending something through the postal service versus email or Skype. We're going to learn what makes some snake venoms lethal and how the anti-wrinkle treatment Botox works. Then you're going to learn about South American arrow poisons and how this knowledge has been useful in modern medicine. You'll also learn how neurotransmitter pathways are altered by addictive substances as well as by addictive behaviors like gambling and even a compulsion to check your phone every few minutes, and how researchers are finding ways to help people break free from the stranglehold of addiction. Let's take a quick run through the nervous system, which for purposes of study is divided into two main parts. The central nervous system, or CNS, is made up of the brain and spinal cord. The peripheral nervous system, or PNS, consists of all the nerves that branch off from the spinal cord and extend to all parts of the body. The peripheral system controls both our voluntary movements and the involuntary movements like breathing and the beating of your heart. The central system is the brains of the operation, guiding conscious and other actions. The central nervous system and peripheral nervous system are, of course, interconnected parts of a functional whole. The brain and spinal cord are connected, so you could think of the spinal cord as being a sort of tail that hangs off the base of the brain and runs down your back, protected by the spinal column made up of bony vertebrae. At regular intervals along the backbone, nerves emerge from the spinal cord. There are two kinds of peripheral nervous system nerves connecting the spinal cord to the rest of the body. One kind, the afferent nerves, bring information in from various parts of the body to the spinal cord. The other, called efferent nerves, carry signals from the brain and spinal cord to the rest of the body. Your 3 pounds or so of brain, your spinal cords, and your nerves are made up of two major kinds of cells. The neurons do the work of receiving and transmitting information. Everything you have ever felt, thought, learned, or sensed, and every movement you've made, every experience you've had, are a result of the activity of your neurons. Then there are the supporting cells called glia, or glial cells, and they don't get much press. In fact, their name is from the Greek word for glue because they hold neurons together. They also play important supporting roles in your neurons. Your neurons are sort of like Don Quixote, making all sorts of dramatic, though not always accurate, decisions about what's what. Glial cells are a companion with a more practical perspective, more like good old Sancho Panza, avoiding trouble, helping take care of injuries. For example, among other functions, glial cells regulate the levels of chemical signals available near neurons. The glia dispose of dead neurons, and the glia even take responsibility for immune defense of the central nervous system. The actual transmission of signals, though, happens within neurons. Let's take a closer look at a neuron. Its main body is called the soma, and it contains the cell's nucleus and most of its other organelles. Bringing input into the cell body are branching structures called dendrites, which are the neuron's receivers, the part of the neuron that receives signals from other neurons. Dendrites on a neuron can get signals from many other neurons simultaneously. It's the combination of those signals that tell the neuron whether it should itself fire and send a signal onward. Stretching away from the cell body is a single axon that carries the cell's output. It's like a long cable that runs from the neuron to the cell it sends information to. The axon running from the spinal cord to your big toe is about 3 feet long, and yes, that's a single cell. Other axons, like some in your brain, may be very short. Short or long, the axon starts from a special point called the axon hillock and carries the outgoing signal. Axons can branch, so a single neuron can send messages to multiple cells. At the tip of each axon branch is a bulbous structure called the axon terminal or presynaptic terminal. These axon terminals are the parts of the neuron that will send signals to the dendrites of another neuron. The axons of some neurons are coated in an insulating layer, the myelin sheath, made by glial cells. The myelin sheath, like the insulation on electrical wires, allows the neuron's electrical signals to be sent speedily and efficiently. What we call nerves are made up of many bundles of axons, piled together into a cable-like structure. To send messages, neurons use electrical signals called action potentials that move down the axon along the soma. These action potentials send signals from the neuron to its own axon terminals. At the axon terminal, the signal must then be handed off to the next cell. This could be another neuron or perhaps a muscle cell. Sometimes the receiving cell is physically connected to the first neuron through what are called gap junctions. Now, these junctions are somewhat misnamed in that they involve direct connections between cells with no gap between them at all. The electrical signal of a nerve cell is directly passed from the originating neuron through the junction to the cell receiving the signal. The sending and receiving cells here effectively function as a single unit. But in many signals passing from neurons, there is a physical gap that the electrical signal cannot cross between the axon terminal of the neuron and the target cell. This gap without a connecting junction is called a synaptic cleft. At this point, the axon terminal will release chemical signals called neurotransmitters that diffuse across the synaptic cleft and bind to receptors on the next cell to pass on the message. The cell on the receiving end of the synaptic cleft is called the postsynaptic cell. Both the electrical and biochemical signaling of neurons depends on the actions of special channels in the cell membrane. These are tiny pores that control the movement of ions like sodium and potassium ions in and out of the cell. Because their function is to regulate ion traffic, these pores are called ion channels, and they open and close as needed. Ion channels are extremely picky. Each one is specialized for a particular ion. A sodium channel, for instance, won't let potassium through. The other thing about ion channels is that opening one is like opening the gate in a dam. Ions rush through the channel at a rate of millions of ions per second. Whether they rush in or out depends on which side of the membrane has the higher concentration of the ion. Just as water always flows downhill, ions move down concentration gradients. To regulate the flow of ions across membranes, channels have gates. Now remember that membrane channels are made up of proteins, so the gates open and close as proteins making up the channel change in conformation. Ion channels involved in neurotransmission have gates that are opened by either an electrical signal or the binding of a molecule. Electrically controlled channels are called voltage-gated ion channels. They open in response to voltage changes across a membrane. Channels that open when a specific molecule, called a ligand, binds to it, are called ligand-gated ion channels. The molecules that most commonly control ligand-gated channels are neurotransmitters. Now, whether they are voltage-gated or ligand-gated, ion channels cause major changes in the concentration of ions within cells when the channel opens. This changes the relative amount of an ion on the two sides of the membrane. For example, sodium ions are normally more concentrated on the outside of the cell membrane than inside the cell. As we saw in the lecture on cholesterol membranes and lipoproteins, this difference is maintained by a cell protein that pumps 3 sodium ions out while pumping 2 potassium ions in. Since the number of ions and electrical charge outside of a membrane do not equal the number of ions and charge inside, the membrane is said to be polarized. This charge difference across the membrane is called the membrane potential. And when no signal is being transmitted, the membrane potential, resting potential as it is called, has a negative value. Generation of an electrical signal or action potential in a neuron begins with opening of sodium channels in a region of its membrane. When sodium channels open, positively charged sodium ions rush into the cell to make the membrane potentially locally less negative. This partial loss of charge is called depolarization, and when it reaches a threshold, 2 things happen. First, nearby voltage-gated potassium channels open in response to the depolarization. Potassium ions then rapidly exit the cell due to the higher concentration of potassium ion inside the cell than out. Movement of positively charged potassium ions out of the cell begins the return to the resting membrane potential. In recovering, however, the cell overshoots, and when that occurs, all of the ion channels close. The sodium-potassium ATPase system then begins pumping ions and restoring the resting potential. The second event triggered by the initial change in membrane voltage is opening of voltage-sensitive sodium channels in nearby regions of the membrane. This leads to depolarization of the neighboring section of the membrane, which then induces the section beyond it to undergo depolarization, and so on. The action potential propagates down the axon like a stadium wave, where each person repeats the action of the person before. Let's imagine wiggling your toe. At speeds of over 300 feet per second, an action potential from the cell body of motor neurons in your spinal cord to your toe would take 1/100th of a second to arrive at the axon terminal at the end of the neuron. At the axon terminal, the signal will be passed on to your muscles chemically across the synaptic cleft, the space between the neuron and the muscle cell. And that's where neurotransmitters, chemical signals made by the neuron, come into play. Neurotransmitters are small molecules made by neurons to cross the gap of the synaptic cleft. In the absence of a signal, neurotransmitters are stored in small membrane sacs called synaptic vesicles near the axon terminals. The arrival of the action potential at an axon terminal is a signal to the vesicles containing the neurotransmitters to fuse with the membrane and release their contents into the synaptic cleft. The major neurotransmitter used at neuromuscular junctions is acetylcholine. When acetylcholine is released, it diffuses across the synaptic cleft about 1/1,000,000th of an inch to the muscle cell whose membrane has ligand-gated ion channels. Acetylcholine molecules bind to these channels and cause them to open. As before, the opening of the ion channels causes depolarization of the muscle cell membrane. This triggers a series of changes within the muscle cells that stimulate them to contract, causing movement. After acetylcholine opens ion channel gates, it is broken down quickly by an enzyme of the muscle cells called acetylcholine esterase. This shoot-the-messenger action is very important for 2 reasons. It prevents prolonged muscle contraction that would cause the muscle to spasm, and it releases the remnants of the neurotransmitter, in this case choline, into the synaptic cleft for recycling. The choline is taken back up by the neuron and reused to make more acetylcholine. Okay, so muscle movement is dependent on the delivery of neurotransmitters from the axon of a neuron nearby. Anything that blocks this neuromuscular signaling pathway blocks muscular contraction. And that's what the neurotoxins in the venom of some snakes do. Muscle paralysis following snakebite is common. Some snake toxins block the acetylcholine receptors on muscle cells and prevent transmission of the nerve signal. Other toxins destroy the acetylcholine esterase that breaks down acetylcholine after it acts to prevent prolonged muscle contraction or spasm. Not allowing acetylcholine to bind receptors prevents nerve signal transmission. And if acetylcholine cannot be disposed of after signaling, muscles remain in a state of continued contraction called tetany. Unless the victims of snakebite receive prompt treatment to counter the effects of such neurotoxins, they will die when the toxins immobilize the muscles involved in breathing. Other snake venom toxins prevent the release of acetylcholine from the axon by entering the neuron and breaking down proteins necessary for synaptic vesicles to fuse with the cell membrane, so vesicles cannot release the acetylcholine into the synaptic cleft. Lacking a signal, muscles cannot contract and therefore remain immobile. Blocking the acetylcholine signal is how Botox injections work to smooth wrinkles. Botox is not snake venom, but is produced by a bacterium, Clostridium botulinum. The botulinum toxin is one of the most potent nerve poisons in the world. Very dilute formulations of the toxin are used in Botox, and they block nerve signaling to facial muscles near the site of injection. Lack of nerve signals prevents muscles from contracting and thus prevents skin from forming wrinkles. But inhibition of muscle contraction is also responsible for the lack of facial expressions, sometimes occurring after Botox treatment. Deadly neurotoxins are not made only by bacteria and snakes. Even plants get in on the action, reminding us that natural does not necessarily mean benign. As early as the 16th century, Western explorers in South America reported native tribes using a plant extract to poison their arrows. The poisonous concoction, called curare, paralyzed animals hit by arrows coated with it. Analysis of the extract revealed several neurotoxins, including one, D-tubocurarine, that was the most effective at causing muscle paralysis. Tubocurarine was of great interest in the early 20th century to surgeons who were dependent on large, potentially dangerous doses of chloroform or ether to knock out their patients. Being able to cause temporary muscle paralysis can be useful surgically, and tubocurarine was the first surgical neuromuscular block. Unlike snake venoms, it binds reversibly, so effects could be reversed after surgery. Understanding how curare worked led to the development of improved neuromuscular blocks, which are used together with general anesthesia during surgery today. So neurons communicate with muscle cells using neurotransmitters, and when that signaling is blocked, problems can arise. Neurons can also communicate with each other. The axon of the neuron that is sending this message releases neurotransmitter molecules. Recipient neurons have receptors that bind those signals and initiate changes within them. In the brain, large networks of neurons communicate with one another and with the spinal cord and nerves to control the activities of the body. Neurotransmitters are central to all of these activities. These signaling molecules work to promote or prevent the production of an action potential in the cells they affect. In the case of neuromuscular signaling, we saw that acetylcholine acted to stimulate the production of an action potential in the muscle cell. Neurotransmitters that enhance production of an action potential in its target cell are called excitatory. If the binding of a neurotransmitter to its receptor stops creation of an action potential in the target cell, it's called inhibitory. Now, glutamate, an amino acid important in nitrogen metabolism, is also the major excitatory neurotransmitter in the central nervous system. The main inhibitory neurotransmitter is gamma-aminobutyric acid, or GABA. A given neurotransmitter may function as excitatory or inhibitory, depending upon what kind of receptors it binds to. Acetylcholine is excitatory when binding to skeletal muscle receptors, but is inhibitory when binding receptors on heart muscle cells, where it slows down heartbeats. Some neurotransmitters affect the way we see the world. Serotonin is popularly associated with happiness and well-being. The vast majority of serotonin in the body is produced and acts in the gastrointestinal system, where it is necessary for normal gut function. Pathogenic amoebae producing their own serotonin can cause diarrhea in humans. But neurons also produce serotonin in the central nervous system, where it positively affects mood, appetite, and sleep. Serotonin is also involved in learning and cognitive functions and is a target of action by pharmacological antidepressants. Another important neurotransmitter in the central nervous system is dopamine. Dopamine is commonly referred to as the feel-good chemical due to a pathway called the mesolimbic dopamine system, the most important reward pathway in the brain. Dopamine's normal function is to make you associate good feelings with things like eating, sex, and social interactions necessary for survival and reproduction, so that you keep doing them. Dopamine may also be described as brain food, given how a deficiency of it appears to play a role in Parkinson's disease. Neurons that produce dopamine in a section of the midbrain die when clumps of a small misfolded protein called alpha-synuclein form inside the neurons. This is when well-known symptoms of Parkinson's disease appear, such as shaking, rigidity, and difficulty in walking. When you do something good for your survival or propagation of your genes, neurons in your brain's mesolimbic dopamine pathway release a little bit of dopamine. But these are also pathways that get hijacked in drug addiction and in behaviors like compulsive gambling. The 2013 update to the Diagnostic and Statistical Manual of Mental Disorders added gambling disorder to the same category as substance abuse. Smartphone use and video games may also activate the same pathways. Drugs can enhance activities of reward circuits by inducing neurons in the brain to release large amounts of dopamine. Amphetamines work this way. Other drugs like cocaine act by inhibiting removal of dopamine from the synaptic cleft. Dopamine release may also be stimulated by nicotine and by opioid drugs. Each mechanism increases the level of dopamine way more than the small natural increases of eating a good meal, for instance. This originally led scientists to believe that dopamine increases were the highs that drug addicts sought. But researchers now think that the actual feelings of pleasure result from release of other neurotransmitters in different parts of the brain following dopamine releases. Instead, dopamine appears to be involved in setting up a conditioned craving for pleasure. Repeated use of the drug and repeated floods of dopamine stimulate downstream pathways and lead to a learning process. We don't fully understand how dopamine-driven learning works, but repeated communication between neurons do result in physical as well as biochemical changes in the brain. And because the amount of drug-induced dopamine is so much larger than the brain's normal little spurts, the brain responds by making fewer dopamine receptors, helping to reduce the effects of a flood of dopamine. But a dialed-down level of dopamine binding creates other problems. The downstream pleasure circuits don't get stimulated to the same extent, so the same dose of drug will no longer produce the same effect. Meanwhile, even though the drug may no longer provide the pleasure it did, drug craving then begins in the rewired brain, leaving the user unsuccessfully trying to achieve the original high. Meanwhile, the normal, smaller elevations of dopamine from everyday feel-good activities no longer satisfy. This makes addicts lose interest in things that once mattered to them, while drug cravings drive them to focus on obtaining the drug. A variation on this theme leads to drug dependence on opioid drugs and prescription painkillers like oxycodone and fentanyl. These drugs bind to mu opioid receptors that are normally bound by endogenous opioid molecules made by the brain. Mu opioid receptors help sense harmful things like pain. Opioid drugs work by binding to those receptors and blocking their action, while also leading to release of the feel-good neurotransmitter dopamine. Binding of the natural ligands to the opioid receptors activates the enzyme adenylate cyclase in the neurons. Now, this enzyme, as we learned in the previous lecture on cellular signals, converts ATP to cyclic AMP. High levels of cyclic AMP in neurons trigger release of norepinephrine, here acting as a neurotransmitter. Norepinephrine as a neurotransmitter functions to maintain alertness, readiness for action, and normal levels of respiration, and it plays a role in the brain's response to stress. In this capacity, it increases breathing, blood flow, blood pressure, wakefulness, and levels of blood glucose. When opioid drugs bind to receptors, they inhibit the activity of adenylate cyclase. As a result, less cyclic AMP is made and little norepinephrine is released. Lack of norepinephrine leads to drowsiness and shallow breathing. This is the reason opium has been used for centuries to help people sleep. If the drugs are used repeatedly, their continued binding to the opioid receptors causes the neurons to attempt to compensate by making more adenylate cyclase. This results in normalizing release of norepinephrine so that the user is alert even after taking the drug. At this point, if the drug is withdrawn, there is nothing to inhibit the adenylate cyclase. But the neurons still have the extra adenylate cyclase, so they pump out excessive amounts of norepinephrine. The result: anxiety, hypervigilance, jumpiness, and muscle cramps. Treatments for opioid addiction have had mixed success. Methadone binds to opioid receptors and stimulates dopamine release just like the drugs it replaces, but it seems to do so without some of the downsides of the opioid drugs, and it alleviates cravings. Naltrexone, another treatment for opioid abuse, is also useful in treating behavioral addictions like gambling. Naltrexone binds to opioid receptors tightly, blocking the binding of these drugs, and does not itself stimulate any pleasure centers. By blocking the binding of drugs to receptors, it prevents the user from experiencing rewards from drug use. But as for all the treatments, getting the addicts to regularly take the medication is challenging. Researchers have observed that regions involved in risk assessment and inhibition of impulsive behavior in the cerebral cortex of users show low levels of activity, so their decision-making can be impaired. This has suggested a possible remedy. Activation of the sluggish region by electrical stimulation has been tested as a treatment for several addictions since 2016. Experiments in cocaine-addicted laboratory rats have shown that electrical stimulation of the corresponding region of their brains using light-based techniques seems to relieve them of their addiction. Transcranial magnetic stimulation is another non-invasive means of brain stimulation with the same aim. It has been tested as a treatment on individuals with cocaine, gambling, and eating disorders. Studies are ongoing, but some preliminary results look promising. The cocaine study used repeated magnetic pulses that altered the electrical activity of the region of the prefrontal cortex involved in impulse inhibition. Of all participants, 67% of the group receiving this treatment shook the addiction, compared to 23% in the control group. If these results hold up in larger studies, they offer hope for many millions of people who would like to kick their habit. Our bodies are wired with incredible nerve circuitry, just like houses are wired to provide electricity to lights and all the conveniences of modern life. And like the light switches and other controllers for the electrical circuits in our houses, nerve cells are activated by electrical signals from tiny neurotransmitters. Neuroscientist researchers elucidate the marvels of the body's cellular circuitry with findings that help keep our biochemical lights on at all times. This is an electrical bill well worth paying. Next time we'll see how our neural wiring gives rise to the 5 senses.